Guys, today we will discuss about the differential staining and what are the difference between the differential and the simple staining. And also, class, we will know the example and method of the differential staining. Okay, class, what is differential staining? Uh, is a specific type of staining that allows for microbe identification and distinguishing between a cells and a mixed sample. So meaning, as differential staining is a staining process uses more than one chemical stain use. So first, utilize a more than one stain, a primary stain and secondary stain. Okay class, we all know the differential staining. So distinguish cells in a mixed sample. So difference between the differential staining and the simple staining. So this is a very different from the simple staining techniques that simply add color and contrast to slide. Simple staining is involved in adding basic cationic dye to the organism. Information of cell size, shape, cell or arrangement also get information about the different features most common use in the laboratory. Okay class, so cationic dye is a specialized dye that use staining. To sum it up, allows the difference to between organism or between the parts of the same organism to be observed. Okay class, there are the methods of the differential staining. So there are the four steps. So first step one, a heat fixmer is a cover with a basic violet dye. So example class is a crystal violet. This is a stain impart is colored to all cells. It is a referred to as a primary stain and apply first. Class, nakoy ipakita na picture diri class para makita jud ninyo class. So mo proceed ta sa step 2. So step 2 is after a short time, the slide is washed off and the smear is covered with the iodine and a mordant. And at least the stage both gram positive and gram negative bacteria appear a dark violet. So step 3. So next, the slide is a decolorized with the alcohol or an acetone alcohol solution. This solution is a decolorizing agent which removes the primary stain from the cells of some species but not from others. Okay class, this is the last of the step of the differential staining. So the slide is immediately washed after a decolorization and the slide is then a counter stain with a basic fuchsin or safranin, a basic red dye. And the smear is washed again, blot dry, and examined under the microscope. Okay, class. There are the example of the differential staining. So there are the five example of the differential staining. First is gram staining and acid fast, capsule and endospore and the flagella. So first is gram staining. Most commonly, use a different staining separates a bacteria cells into two groups based on the cell wall structure. So gram positive and there are the gram negative. So ang gram positive class is a steep purple on the staining. So take ang 52 glycone cell and it is a special carbohydrate. So and also and gram negative then a 52 glycone cell and outer membrane present and stain result pink. And the second is acid fast staining. So differentiate between the cells that have a mycotic acid and the present in their cells in the cells walls, and these that do not acid fast and contain a mycotic acid, and the non-acid fast do not contain a mycotic acid. And the third is a mycotic acid is a waste to lipid. And this there are the third example. So capsule staining and located outside the cell wall bacteria and distinguish between cells that have a capsule present and those that do not and do not utilize a negative stain and capsule one protective structure or prevent a dying and prevent phagocytosis. Endospore stain perform to detect the presence of endospores. These are special structure produced by a certain general bacteria such as genus Bacillus and genus Clastodia, and endospores dominant structures produced by some species in harsh environment. So serve to protect the DNA of a species. Okay ma'am, so to sum it up, differential staining is a process to differentiate cell structure, size and shape. It also give gives deeper information and about cells, and it composed of the four step of the methods process, and it also and have five examples, namely gram staining, so acid fast and capsule and endospore staining. And I hope that you can learn something, ma'am, about this my report, ma'am.